So then I would like to introduce Professor Bing Ren. He's a professor of cellular and molecular medicine in UC San Diego and the director of UCSD, the Center for Epigenomics. So thank you very much for joining today.
Good morning, everyone. It's my real pleasure to join this exciting conference. Just before I begin, I want to make sure I'm sharing the full screen with you.
Yes, indeed.
Yes.
Looks great.
Great,
Yes.
and you can hear me. Great, thank you.
Yep.
I want to thank this organizers, Peter and others, for giving me this chance to join this Chromatin-Con virtual symposium on aging, a very, very interesting topic. I have some disclosures to make. I'm a co-founder of Epigenome Technologies and Arena Genomics. And the topic of today is about epigenetics and aging. And I want to begin by showing this picture that depicts the first emperor in China looking for a way to extend his longevity, and he went such extends length by sending 3,000 young men and young women to the East China Sea to look for recipes for extending longevity. And that was 2,000 years ago. Of course, he died long before anything was discovered. It just goes to say that understanding why people age and how to modulate that has been a ancient concept and continue today. And in this conference, I think epigenetics has been mentioned multiple times. And I like to mention also that epigenetics refers not only just to DNA methylation, but also to a collection of chemical changes to histone proteins, such as acylation on histone proteins and methylation on different lysine residues. And this collection of chemical modifications to DNA or the protein are known as epigenetic regulation. We already heard that epigenetic DNA methylation patterns can predict chronological age and has been widely used for connecting the epigenetic modifications on DNA to the aging process. But there has also been finding that other modifications to the genome also correlate with age. In fact, loss of hydrochromatin has long been proposed as a mechanism of cellular senescence and aging. This has been first reviewed in yeast, but also later in human cells, human conditions, premature aging conditions, for example. And there has been extensive literature on this very topic. And the hydrochromatin mainly refers to the formation of a chromatin structure due to the primethylation or dimethylation of lysine 9 on histone H3 that would recruit repressors and form compact chromatin, allowing preventing gene activation in that region. There's another type of hydrochromatin known as polycomb that can also contribute to formation of gene silencing. That the relationship between polycomb and the gene regulation has not yet been well established. More recently, it has now shown that loss of, it has been proposed that epigenetic information is lost, and that is also a base of aging. There's a very recent paper published a couple of weeks ago in my Sinclair lab that beautifully illustrate this concept. I think the main reason people began to take a look at epigenetic mechanism for aging is that epigenome, unlike DNA sequences, can be modulated, is reversible, and this provides a window of opportunity to perhaps intervene and reverse age-associated declines in cellular and tissue functions. But there's still a significant knowledge gap, and we, like our previous speaker touched upon, it's still unclear how epigenetic changes contribute to aging process. Is it cause, is it passenger, and what is the true mechanism, how that they might be related to age process. And to address this, we need to identify the genes and the cell types that show age-associated epigenetic changes and to study how these changes are linked to aging process. And I'd like to introduce a few techniques that we use to profile the cell's epigenome. Very early on, when I started my lab, as the inventor of the chip-on-chip technology, that was before DNA sequencing was invented, high-throughput DNA sequencing was invented, and others are using this method to profile locations of histone modifications in the genome. And quickly in the last 15 years, now we have been using a combination of a chip or tag or DNA's hypersensitive mapping and whole genome bisulfite sequencing with next-gen sequencing techniques to provide detailed maps of histone modification and DNA methylation patterns, as well as open chromatin along the genome. This allows us to define regulatory elements such as enhancers and promoters and define the state of activities of such regulatory sequences. And more importantly, when we use a method called Hi-C, we can determine the higher order chromatin structure, linking distal elements to their target genes. So all these tools have been now widely used to allow us to study gene regulation and, more importantly, analyze the dynamic epigenome landscape in cells and during development and during aging. Because epigenome is highly variable, highly heterogeneous between cells, between cell types, and between tissues, it's very important to also use single cell techniques to resolve the heterogeneity of epigenome among a tissue. And technologies have now been invented to assess the various epigenetic marks in single cell level. For example, a few years ago, we have adopted a method to do whole genome chromatin accessibility assays by combining PM5 transposes with high throughput split and pull combinatorial indexing to study the open chromatin in individual cells. And we made this procedure highly robust and automatable. And this has actually been a major approach that my lab used to study epigenome diversity across tissues and cell types. Another tool that we contributed to the field is single cell methylation combined with Hi-C analysis. This allows us to map both the methylation patterns and the chromatin conformation in individual cells in parallel. Having methylation allowed us to extract, to cluster cells based on the similarity of methylation and to extract then chromatin conformation in individual cell types. And finally, we also recently developed a method that allows us to investigate histone modification as well as transcription genome-wide so that and the individual cells. This again called pair tag allow us to use cut and tag approach invented by Steve Hanikoff combined with combinatorial indexing strategy to then study DNA, histone modification together with gene expression from the same cell. I'm going to describe how we use this method to study aging process. I want to introduce a brief strategy that how people take single cell data to study epigenetic processes. As many of you are aware, single cell technologies all suffer from one issue, that is the data sparsity. So in the beginning, there's a lot of doubt on how such sparse data could actually reveal important biological processes. And to overcome this limitation, a computation approach known as clustering, it was developed to group cells based upon how similar they are. And such similarity typically is rooted in the fact that cells of the same type typically share a very similar epigenome. And consequently, using computational tools, you can basically group individual cells, sparse single cell epigenome data sets into cell clusters. And now you can perform pseudobulk analysis, aggregating the epigenetic data signal from individual clusters, treating them as a cell type specific epigenome maps. And this then allows us to study epigenetic processes in complex tissues. So what I'm going to describe today is a recently published work that we did to use epigenome analysis to study aging process in mouse tissues. This was carried out in collaboration with Dr. Xiangying Xu from University of California in Irvine, and work was done at UCSD. Sebastian Prizo led the data generation effort. Yan Xiaozhang and Luisa led the DNA data analysis, and they were also assisted by Chen-Shui Zhu, who is now at New York Genome Center. So this study basically focused on 5 tissue types, including frontal cortex, dorsal hippocampus, leg muscles, bone marrow, and heart, and then study how cells in each tissues change the epigenome in the course of aging, from 3 months, 10 months to 18-month-old mice. These are all male mice. We currently focus on the male in this stage, but we are also carrying out similar analysis using female mice. But today, the result I'm going to present you are focused on the male. We perform single-cell TAC-seq, RNA-seq, and also combine the histone modification and RNA analysis to study how epigenome changes in the course of age in different cell types and different tissues. So it's probably not very clear to many people that mouse age at considerably accelerated rate compared to humans. And there is a rough correspondence. 3-month-old mouse is roughly like a teenager, and 10-month-old mice are more like middle-aged, and 18-month-old mice are more about the beginning of the old stage. So they're not super old. We pick the 3 time point just to see, give us a chance to see whether there are age-dependent changes in different cell types and tissues. Altogether, we carry out single-cell TAC-seq analysis for 227,000 cells across these 5 tissues, across 3 different age groups. Each tissue we analyze in duplicates. So that's the total number of cells we profiled, and we then performed clustering analysis. This resulted in the identification of 32 major cell types that encompass lymphoid, myeloid, neurons, excitatory neuron, inhibitory neuron, for example, glial cells, and muscle cells, and endothelial cells, fibroblasts, for example. When we look at the cellular composition of different tissues across the age, we don't see dramatic changes during aging. This is similar to what Ashley talked about yesterday. This is two examples, dorsal hippocampus and bone marrow. This is corresponding to the different cell types and their fraction in the tissue. Basically what was shown are two replicates of each age group in each cell types. And you basically don't see consistent and robust changes for essentially for all of them. There are changes in chromatin accessibility that we can detect. We found a total of 77,800 candidates cis regulatory element out of almost 350,000 that we identified from the collection of cells that show differential accessibility between 3-month-old and 18-month-old mice in at least one cell type. And age-associated changes in chromatin tend to be restricted in specific cell types, indicating that these are probably related to specific functions of that tissue or cell type. It goes without saying that when we do see accessibility changes, they tend to correlate with transcriptional changes nearby for the nearby gene. These are a few examples. ROBO1, for example, are shown age-dependent decline in accessibility, and that is correlated with transcription of this gene during aging. And same, the reverse is true for ITGB5 and so on. So when we have these patterns, typically you could do motif analysis to ask what transcription factors are related to the changes. But one thing we, Yan Xiao Zhang performed is to compare the accessibility dynamics with previously annotated epigenetic regions in the mouse genome. Specifically, we compare the we compared for each cell type, the age associated the reduced accessibility or increased accessibility with regions that are marked by 6 different changes, epigenetic changes, including lysine 9 trimethylation, a hydrochromatin mark, lysine 4 trimethylation, a promoter mark, and so on. And one thing that stood out was that when we look at the excitatory neurons, across all excitatory neurons, the age-dependent gain of accessibility all show very strong enrichment at hydrochromatin mark, lysine 9 trimethylation that was detected from bulk brain tissues at birth. And this is present only in the excitatory neuron, but not in inhibitory neuron or glial cells. And this p-value is highly significant and the odds ratio is very high. And that immediately raised the possibility that excitatory neurons are losing hydrochromatin as a result or to accompany the gain of accessibility in such regions. And looking at one example, this is indeed the case. For example, in this hydrochromatin domain marked in blue shade, you can basically see in several types of excitatory neuron. These are layer 2, 3 in the frontal cortex. You basically see at 3 months, there's very little accessibility, but from 10 months to 18 months, their accessibility is significantly enhanced. Same is observed for layer 4 excitatory neuron in the cortex. But you don't see this pattern in the oligodendrocyte cells or a inhibitor neuron called a BIP in the same tissue. And you can see the same trend in another region in the genome. So we hypothesize that the gain of accessibility in these regions might be due to loss of hydrochromatin. But how do we prove it? And that's where Chen-Xu Zhu, who invented the tool called paired tag, come into help. So paired tag, as I mentioned, enabled us to interrogate both histone modification and RNA expression from the same individual cell at very high throughput. So we performed paired tag against frontal cortex and the dorsal hippocampus for 3-month-old mice and 18-old mice. And this is a procedure to describe how paired tag is worked. Because this work is published, I won't provide more details of it. We performed paired tag for each tissue in duplicates and generated approximately 20 to 30,000 cells worth of data, and we grouped them together with the first of all, we clustered them based on RNA expression patterns, and we were able to annotate different cell types such as CA1, CA2, 3 neurons, and the dorsal DG neurons from hippocampus, and the cortical neurons here, and the hibberellin neurons. So we then ask, does heterochromatin really get lost during aging process? And this is a few examples I'm showing you to confirm that this is indeed the case. So what you are seeing is this track. This corresponds to lysine trimethylation. So the blue corresponds to 3-month-old, and the red corresponds to 18-month-old. And you see indeed there is a quantitative loss of heterochromatin signal from the young to the old mice. This is true for all the excitatory neurons, but not true for inhibiting neurons shown down below, or astrocytes or oligodendrocytes shown up here. You can see this is pretty consistent across other heterochromatin regions. And when we perform this against all domains that was previously annotated, we see that a ataxic signal is strongly induced or gained in the excitatory neuron class, very rarely in the other cell classes. And correspondingly, we also detect transcription of genes to be strongly enhanced in a subset of this heterochromatin regions. And by looking at the paired tag data, we can see quantitative loss of lysine trimethylase signals for many different classes of heterochromatin domains. So by and large, we confirm that there is a dramatic loss of lysine trimethylation in the genome across known lysine heterochromatin domains. This is associated with DNA gain of DNA accessibility and transcription activities during aging. But this is only found in the excitatory neuron and not in other cell types. And that is the interesting observation. One class of elements that are gaining expression or transcription is L1 elements. In fact, L1 element is known to be silenced by heterochromatin. And we suspect that L1 expression is enhanced during aging. To confirm that, Dr. Xiaoming Zhu's lab performed immunostaining using antibodies against L1 ORF1. And basically you can see that in the, this is in the frontal cortex, the L1 ORF1 is rarely detected, but in 18-month-old mice, their protein levels is quite widespread. So to know what type of cells are expressing L1, we basically use CAM kinase 2 alpha as a marker for excitatory neuron. You basically can see that the CAM kinase 2 staining is always accompanied with this increased L1 ORF1 signal patterns. This is quantification. Basically, you can see the intensity of ORF1 is enhanced in the during aging. So what this observations indicate is that chromatin accessibility measure allow us to interrogate open chromatin and dynamics in multiple cell types and tissues during mouse lifespan. We observed dramatic loss of heterochromatin during aging that was restricted to the excitatory neurons in the cortical or hippocampal region, hippocampus. And there are multiple classes of repetitive DNA elements that become de-repressed during aging in these excitatory neurons. The question is, how does this loss related or even contribute to aging? And if so, what's the mechanism? Here, we're still performing additional analysis. I just want to share with you some thoughts. So I want to remind you that about 6-7 years ago, a study from John Saddery group at Brown showed that L1 transcription become activated during senescence in fibroblasts and can indeed drive interferon response in these cells to promote age-associated inflammation. And that certainly can be a mechanism for loss of heterochromatin to contribute to age-dependent phenotypes. And also just a few months ago, last August, a study from Valeria Orlando and Balmente lab actually show that L1 RNA expression likely contribute to heterochromatin loss in excitatory neurons. Sorry, not excitatory. In their study, they did this in a mouse, in whole mouse. What they show is that you can suppress L1 RNA, and that can actually extend lifespan in progenitor mouse models of progenitor, a premature aging model. So we suspect, based on our data, that perhaps L1 RNA expression that accompany with the loss of the chromatin in excitatory neurons could also contribute to this process. And just a week ago, another paper from in this publishing Cell from Guanghui Liu's lab in China showed that another class of endogenous retrotransposal elements known as ERK are activated in aged cells in humans, and this can trigger an innate immune response and cellular senescence. ERK is one of the youngest class of endogenous retroviral element in the human, and it's actually can still be shown by Joanna Vaseka many years ago. You can still form viral particle variants, and that can actually induce CGAS STING pathway to trigger inflammatory response in the cellular level. And this potentially is a mechanism for aging, and also pointed to potential pathways to inhibit such reactivation of retrotransposal elements to serve to potentially alleviate age-related phenotypes. Now I want to share with you some ongoing study that actually provide even more insight into the age-related process. This study is performed in collaboration with Joe Acker and Margaret Barron from Salk Institute. Here we focus exclusively on the mouse brain, taking advantage of our previous studies on this frontal cortex and the hippocampus, but now extended to 8 total brain regions that cover from the anterior to posterior, and essentially most of the mouse brain areas. So we again perform single cell TAC-seq and in working at Joe Acker's lab performed single-cell methylation studies. And in this process, we analyzed a total of 600,000 cells. This allows us to group them into 57 major cell types. So this study provided really in-depth view of the changes of open chromatin in different brain cell types, again, much more depth over what we previously performed that I showed you earlier. Again, we grouped this different cell classes into exciting neurons, inhibiting neurons, and glial cells. And the brain distribution is shown here. And for each age group, the color indicate for each cell cluster, there is a general nice mix between the young and the old mice. And having this in-depth and a large number of cells really allow us to ask if in the rare cell population, such as progenitive cells, we do see age-dependent decline in cell abundance. And the result is pretty clear. For example, immature oligodendrocyte fraction do show age-dependent decline in all the brain regions that we analyzed. For example, here the dark blue are 8-week-old mice, and the gray and the yellow, you can see that very early on, the fraction of such progenitor cells decline. And these progenitor cells are responsible for myelinating neuron axons. So without many of such progenitors, you can imagine that myelination repair might actually be at risk. Another class of progenitor cells that show age-dependent decline are the radioglial-like cells. These are, for example, this is shown here in this is the radioglial cells. So in several brain regions, they do show age-dependent decline, in particular in nuclear accountants and in this is the PAMM. So these cells are actually interesting because they are responsible for adult neurogenesis, and they are important for the replenishment of damaged neurons or astrocyte. So you can largely suspect that such cells loss might impair the ability for adult brain to provide a renewed function when there is a damage. So these are very preliminary data. I'll be happy to answer more questions on them, but I want to stop and also acknowledge my colleagues. I mentioned their names while I was speaking about their findings. I want to particularly mention the work done at UCSD's Center for Epigenomics. This is a co-laboratory that provide single-cell epigenomic assays and spatial imaging and high-throughput epigenomic assays in a collaborative fashion with faculty at UCSD and beyond, and also have extensive collaboration with industrial partners. I mentioned my collaborators, Xiangming Xu, Margaret Barron, Joe Acker. I didn't have time to talk about our ongoing work with Peter Adams as part of the CNAT project. Thank you again for your attention. I'm happy to address questions.
Thank you very much for a great talk. You can see already several questions and also between our panelists. So maybe I will start with Vera. She has been very, very fast with the questions.
Thank you. Thank you being, really tour de force. So I have a question. When we look at aged mice and we see line 1 reactivation in various tissues, like lungs and skin and intestine, with the corresponding interferon induction. So I'm curious, why could it be that in your data set, you only see it in excitatory neurons? Is there anything that, it doesn't meet the threshold of detection, or like what could be the reason that it's just those neurons?
I want to clarify one thing. We saw loss of heterochromatin that correlated with activation of line 1, but we did not see loss of significant loss of heterochromatin in other cell types. But heterochromatin include not only line 1, but also other classes of endogenous retroviral elements or just simple repeats. So it is actually more broad. So I won't equate line 1 to heterochromatin loss. There actually, let me show you one more example which actually support your study, support your, sorry, did I, okay. So in this slides, actually, we do see, like what you said, a enhancement of L1 or F1 staining in nonneuronal cells. How this came about, we don't know, because from heterochromatin loss analysis, we don't see significant loss in the heterochromatin, but for some reason, we do see enhanced staining even in non-excitatory neurons. And I'm puzzled by this, and I'm actually not sure how to make sense of it. But what we do see is a much stronger enhancement of line 1 signal in the excitatory neuron.
Thanks.
Thank you. Then I think I'm going to take one of the questions from the Q&A. So it's from John Puch, and he's asking, are there metabolic features of excitatory neurons which distinguish them from other cells? And if so, if this might be interacting with the epigenome?
Yes, this is a very good question, and I've been asked multiple times. We have some sense on why this excitatory neurons might actually be more prone or more vulnerable for loss of heterochromatin. It is when we look at excitatory neurons, these are the ones that actually already have a modest level of accessibility at the heterochromatin. And almost a decade ago, Rusty Gates lab actually showed that neurons can actually have reactivation of line 1 elements just in normal process. So we actually have we found that line 1 is prone to be active even at young mouse in the excitatory neurons. And that is probably separating these neurons from other non-excitatory neurons. And I have a hypothesis that perhaps this is a feedback loop. When you have more line 1 expression, you tend to lose more heterochromatin. And we're still, we don't have yet proof on this yet, but that's my suspicion.
Thank you. I think in line with that question, so there is an anonymous attendee that is asking, it seems that unlike excitatory neurons, glia upregulates histone 3K27 acetylation and histone 3K27 methylation, 3 methylation marks. Does that represent a glial response to loss of heterochromatin in those neurons?
That's a very good idea. We suspect that this is a process, for example, activation of line 1 may perhaps trigger inflammatory response, and that perhaps leads to cell-cell communication, where basically the secretion of the cytokines and leading to other cells, especially glial cells, to attack them. So that is what we suspect happening too. Needs to study it in detail.
Thank you. I think if we have time, then I can continue with a couple of questions more from the Q&A.
Yeah, absolutely. I was thinking we go to about 1/2 the hour, and then Rong Fan is ready to go when. And Perfect. It looks like there's one from Gary in the chat too, if you need another question.
Didn't see it from the chat, sorry. Yes, here it is. So I will read that one from Gary. Does the reactivation of line 1 lead to genomic instability in the excitatory neurons?
It's possible. I think line 1 encode 2 ORFs. One is ORF1, ORF2 actually encodes a endonuclease activity, and that is likely, if it's indeed overexpressed, leading to double-strand breaks in the genome. So I would highly suspect this is happening. Yeah, we're certainly interested in looking closely into that.
Yeah, thank you. So I have another from Sarah Frias. She's asking if is it possible that the reversal transcriptase might become more active when the heterochromatin is lost?
That's what I also suspected. As I said, line 1 ORF2 has both has an endogenous endonuclease activity, but also has a reverse transcriptase activity. So therefore, it's very possible that when you have high levels of line 1 transcription and protein levels as detected by the antibody staining, it's very likely that they can lead to higher levels of reverse transcriptase.
Mute. Hi. Good. So I have another question here with the raised hand from Gary.
Okay, I'm just going to try and see if the audio works. It seems to. Hi, Bing.
Hello.
Hello. My question is actually a conversation just with Joe Fernandez on hydroxymethylation in the brain. Have you plans to look at that as well?
Yeah, hydroxymethylation is an intermediate of DNA methylation. So yeah, it's a very interesting mark to study because it typically represents regions that are in the process of undergoing erasure of DNA methylation, which typically happens at enhancers. So it's very interesting. It will be very interesting to look at that. I think people are still developing method to study hydroxymethylation in single cells. I know some methods that have been developed to address it, but how general it can be carried out at scale, I think this still requires some time.
Yes, and I just know that it seems to be more prevalent in brain tissue. Do you have any thoughts on why that is? Is there a function associated, or is it just due to?
Yeah, yeah, that's an interesting question. I actually don't know. I can guess. It may be that brain has a more I don't know, more more DNA methylation that has to be removed or due to some processes. So I'm not sure I have the answer actually.
think there is one last question from Peter.
Hi, Bing. Great talk.
Hello.
So I was thinking about the discussion you had with Vera earlier about, you know, the apparent inconsistency, I guess, with some of her data, and also, you know, in your data in terms of, you know, heterochromatin changes versus line expression. So in John Sedevy's paper that you mentioned, I mean, he proposed that there were multiple mechanisms for regulation of line elements in senescent cells. I think loss of RB, inactivation of the Trex nuclease, and also I think activation of one of the FOXO transcription factors. So I wonder whether, you know, perhaps looking at those additional levels of regulation could shed some light on those apparent inconsistencies. There may be more to it than simply loss of heterochromatin, and potentially those things vary between mouse strains as well.
Thank you, Peter. That's a great idea, great thoughts. I'll follow up with you on that. If we still have time, I see that Sarah Algin have a question in the Q&A.
Yeah, give me one moment and I can, I'll unmute her. Sarah, I'm gonna allow you to talk here.
Okay.
Go ahead when you're ready.
All right. It looks like some of this might be explained by a failure to recognize double-stranded RNA, a failure to identify those sites for silencing. Has anyone looked systematically to see if there's a disruption in that pathway as a consequence of aging?
Recognize double-strand RNA by the machinery of heterochromatin? Is that what you're trying?
Yeah, if you don't have that mechanism of where to target your heterochromatin formation, you could lose the whole thing. It could start to unravel.
That's very interesting. I haven't looked at that, but that's possible. I imagine that it's much to do with the maintenance of heterochromatin and not how it's established in the first place.
Yeah.
Right, because neurons don't divide once they in adult life. So I suspect this has to do with how they are maintained. But I think double-strand RNA potentially is indeed a possibility in this process during the maintenance process.
Well, if the focus is on maintenance, then the question might be, what do you see in terms of maintaining levels of HP1? That's a marker that some people have looked at. If that's decreasing in the cells and you do not have sufficient HP1 to maintain the stable assembly, there you go.
Very good point. We'll target HP1. Well, thank you. Thank you for that.
Thank you. I think we should move to the next speaker, and thank you very much, Big Ren, for your presentation.
Thank you for your attention.